DNA fingerprinting, using electrophoresis to separate DNA. The first step in running electrophoresis is to set up the electrophoresis chamber and prepare a gel. Small metal dams are placed on each side. A plastic tray is oftentimes placed in the center. Notice that the tray has notches on either end. This is so the comb may be set into place correctly. Always place the comb towards the black cathode so that the DNA will run towards the red cathode. Molten agar is then carefully poured into the chamber. Be careful when pouring the agar not to introduce any air bubbles, and you only want a gel that is a few millimeters thick. After pouring the agarose, the chamber itself should be set aside until the agarose gel has solidified. For this experiment, you will need a micropipetter that measures between 2 and 20 microliters. 5 microliters is the first volume to be used. You should have 4 samples: sample B, sample E, sample H, and patient X. For each sample, you will need to load 5 microliters of loading dye. Loading dye allows visualization of different sizes of DNA bands, although the dye itself is not associated with the DNA in electrophoresis. Glycerol in the loading dye will help weigh down your sample when loading your wells. Your instructor may also add a small amount of cyber green to each of your samples for observation later on. Now that your samples have been prepared, you need to centrifuge each of the tubes. Make sure the centrifuge is well balanced and all lids are replaced before turning it on. Centrifugation should not go above 13,000 RPM. By now, your gel should be solidified. Gently remove the comb and the dams in preparation for pouring buffer. For this experiment, you will be using a 1x TBE buffer. Fill one side of the electrophoresis chamber, and then fill the other, allowing the buffer to just barely cover the surface of the gel. If too much buffer is applied, the electrical current will run over your gel instead of through the gel, and the DNA will not migrate correctly. You are now ready to load your samples into the wells in the gel. Turn the chamber to an angle with which you can easily see the wells in the gel and under the buffer. You will now need to obtain your micropipette and reset the volume to 10 microliters or another volume as given by your instructor. Loading an electrophoresis gel can be as much art as it is technique. The trick is to be sure not to puncture the well and not to put the tip of the micropipetter too far into the well. The glycerol in the loading dye will help guide the sample into the bottom of the well without the need of putting the tip into the well. Hold the micropipetter with both hands and gently push the plunger. Proper placement of the tip ensures that the loading dye will make your sample drop easily into the well. This process should be repeated for each of your samples. Your samples should be loaded in alphabetical order, and one lane should be skipped between nurse H and patient X. This ensures that you will know which sample is in which lane when observing your final results. Now that your samples are loaded into your gel, You are ready to apply the electrical current. Most electrophoresis chambers will only allow you to apply the lid in the correct orientation. Put the lid of the chamber in place and plug it in. Your power supply should be set to 120 volts. Press Run. You can determine that your electrophoresis chamber is working properly by the presence of bubbles at both the positive and the negative cathodes. As the electrical current is applied through the gel, the loading dye will separate into 3 different colors. You should run the electrophoresis until the purple band in the middle is at the end of the gel. This ensures separation of the DNA fragments. After your gel has completed, you should then take a small plastic bag, write your group name on it, and remove your gel. Carefully remove the gel, supporting it from the bottom with all of your fingers. Place it into the bag, and you may then put in your buffer. During the next lab period, you will be observing your gel under UV light. Electrophoresis is an important tool for microbiologists, researchers, diagnostics, and forensics. It is used extensively throughout the world and remains one of the most common tools used in laboratories.